I'm Professor Burgess, and I'm going to show you how to use a microscope. So what we have here is a binocular compound microscope. It's called binocular because we use two eyes, and it is compound because we magnify an image twice. Once using what we call the objective here, where we control how much magnification, and a second time through the actual eyepieces up here called the oculars. The oculars will magnify our image another 10 times. We'll go over that in the lab, but for now I just want to show you some of the basics. So here's our microscope. At the top of the microscope, the eyepieces that we look through with our eyes are called oculars, and again, these will magnify an image 10 times. Through here is the body of the microscope. This section right here is called the arm. Attached to the arm is the stage. On the stage, this is where we will add our slide. Here's a slide, a little slide of a paramecium here. In order to add a slide, I'm just going to tilt this so you can see, we have a clip here. You're going to pull this clip back, put your slide onto the stage, fit it into, there's a little corner here. Here, release the arm. Once you release the arm, that will hold your slide in place on this. There's a hole in the stage which light will shine through, and underneath the stage down here is called the condenser. On the condenser, we'll move this up so you can see it. The condenser here, we control the intensity and we also control the contrast. So there's a lever here to move back and forth. This will control the contrast and the intensity of the light that's coming through. Below that is our light source. This is where the light will be coming from. But you may want to know, how do we control all of this? Well, we control it over here on the side. When we want to focus our microscope, we first put our microscope on what's known as the 4x objective. These objectives here are how we control magnification. The 4x objective is also known as a coarse focus, or a high and dry. We use the coarse focus knob, which is the large one on the side of the microscope here at the bottom. So this knob, as you can see, will move the stage up and down so we can bring our specimen into focus. Inside of the coarse focus knob is the fine focus knob. Once we get our specimen generally in focus with the coarse focus, we can use the fine focus to really kind of fine tune what it is that we're looking for. Once we're in focus with the 4x objective, we're ready to move to the next magnification. The next magnification on these microscopes are called the 10x. This will magnify our image 10 times. When we look at it through these oculars, they magnify it another 10 times. This gives us a total magnification of 100x. So if we multiply the objective magnification times 10, which is the ocular magnification, we get what's known as a total magnification. When we were here on 4x, that was 4 times 10 is 40, for a total magnification of 40x. In addition to our focusing points, there are a few other knobs on the side of the microscope you should be familiar with. Right here is the light intensity knob. If I turn this microscope on, you can see there's a pretty good light source. I can adjust the intensity of the light, how bright it is, with this light intensity knob right here. I can also move my slide around. Sometimes when we're looking for something really small, it may not be immediately where we look, so we have to move the slide around. The top portion of this stage knob here comes down. This will move our stage back and forth so we can look forwards and backwards on our slide. The bottom part of this, if we turn, will move our slide left and right, so we can hunt around and look and try to find what it is that we're looking for. Be sure to take a look through your lab manual. Make sure that you are able to define some of the terms such as parfocal, total magnification. You want to make sure that you're able to calculate a total magnification. I'm sure your instructors are going to ask you to do that. Also, total magnification is going to be used in your lab reports when you report your drawings or whatever images you may have that you may have seen on the microscope. So you have to report total magnification. Objective magnification times your ocular magnification. And for our lab, that's going to be times 10. So objective times 10. You also want to make sure that you understand parfocal. You also want to make sure you understand how to focus the microscope. And of course, be able to label all of the parts. Oculars, arm, stage, slide clip, coarse focus, fine focus, light intensity, condenser, down here at the bottom, condenser. And of course, at the very bottom, your light source. There you go. Those are the basics of the microscope. Enjoy the lab, have lots of fun, and do good on that pre-lab quiz.